In this tutorial, we're going to learn how glucose is measured from the skin based on skin structure and physiology. So let's start with the epidermis. At the top of the epidermis is the stratum corneum. This is these shriveled up little cells that dry and flake off, and that's what faces the outside world. Then we have some other layered structure there. Then we have these epidermal cells, and those are going to be the star of our show later on. We'll talk more about those. And then we have the basal layer. So the epidermis altogether, those layers, is only 100 microns thick. That's not very thick, 1/10 of a millimeter. And the whole thing is filled with interstitial fluid. So all the voids between cells that's inside of our body is filled with interstitial fluid. This is really important to note. Interstitial fluid does the job of transporting nutrients to all these cells and taking away waste material. There's something called histology, which is where they do thin slices of tissue and then look at it in a microscope, and they can actually do quantitative histology, which is where they take images and measure cross-sectional areas. And from those studies, we know that the epidermis is about 70% cells and about 30% interstitial fluid, the fluid between the cells. Next, we have the dermis, and the dermis is quite a bit thicker compared to the epidermis, 1 to 2 millimeters thick, again filled with all this interstitial fluid, and it's supported by all these collagen fibers. So instead of a cell matrix like the epidermis, it has these collagen fibers and elastin, and it makes this really tough layer. But no cells, mostly no cells. We're going to remove that just so we have some space to draw. Now we're going to add the organelles. There's organelles in the dermis, hair follicles, oil gland, sweat gland, lymph vessels, and blood vessels. Those are all structures in the dermis, but it's all structured within this collagen fiber network instead of some cell matrix. If you look at a breakdown of the dermis, the collagen and all the organelles make up about half, about 52% of the volume. 8% of it is plasma. That's in those blood vessels. I've only drawn one here, but there's a whole bunch of them. They're really small. They're not very big, but there's enough of them that you have a significant amount of blood plasma. And then 40% interstitial fluid. So a lot of the space is interstitial fluid, much more than any other parts of the skin. Okay, we're going to leave that up. Now we're going to add the adipose tissue. This is below the dermis. This is the third layer of skin. It's much thicker again. It's 2 to 7 millimeters thick below the level that you can see in this drawing. And in addition to those kind of fatty cells, you have the capillaries which go through them. Those capillaries have all the nutrients in them. They have the blood cells in them, and those feed those little vessels that I left drawn on the right-hand side. Those vessels get fed from the capillaries. A breakdown of the adipose layer is 78% cells, 3% plasma, and 20% interstitial fluid. So still a reasonable amount of interstitial fluid and plasma. The reason I highlight those things is they become important later on. So now we're going to talk about glucose migration. Let's add some glucose. In this case, they're going to be all these little blue dots, not to scale obviously, but we need to be able to see them. We're going to start with a high concentration of glucose in the blood and a low concentration in the interstitial fluid. Well, glucose wants to get from the high concentration area to the low concentration area through simple osmotic diffusion. It just diffuses through the capillary and vessel walls to populate glucose into the interstitial fluid to try to match the concentration. The glucose in the interstitial fluid then can make its way to the cells because the cells get all their nutrients from the interstitial fluid. Once the glucose is in the cells, it's converted to glucose 6-phosphate. It does not stay as normal glucose. It gets phosphorylated. After it's phosphorylated, that glucose can no longer come back out of the cell. It is trapped in the cell until it's consumed by the cell for energy. In the adipose tissue, the glucose is also converted, but instead of converted to glucose 6-phosphate, it's converted to triglycerides. So what is insulin? What happens when when insulin is introduced in the system, either from your pancreas or from an external injection? Let's zoom in on a cell. And now we have the cell, we have its membrane around it, we have these transport proteins, which we'll see them in action in a moment, and inside the cell is glucose 6-phosphate. Well, the glucose outside of the cell in the interstitial fluid sees a low concentration of glucose inside the cell because it doesn't recognize glucose 6-phosphate as glucose. So that glucose outside would want to diffuse into the cell, but it can't get past the cell membrane. It has to go through the transport proteins. So once it goes through the transport protein, it can go into the cell. That process is called the GLUT1 process. And once inside the cell, it again will be converted to glucose 6-phosphate. So very quickly. So the concentration of glucose in the cells is always low because it's converted to glucose 6-phosphate. So now we're going to add insulin. Insulin's a hormone, and what insulin does is when there's insulin in the system, the transport proteins come to the surface and make a whole bunch more surface area for glucose to go into the cell at a much faster rate than it did before. And then again, all those glucoses get phosphorylated to glucose 6-phosphate. Again, always sucking the glucose out of the interstitial fluid because the glucose in the interstitial fluid is always trying to get an even concentration of glucose inside and outside the cell, but it can't because the cell is always converting it to glucose 6-phosphate. So as those cells consume that glucose 6-phosphate from the interstitial fluid, more glucose then diffuses out of the capillaries and the vessels. Osmotic diffusion out of the capillaries and vessels, and facilitated osmotic diffusion into the cells. So some notes from this is just a reminder, glucose is quickly converted to glucose 6-phosphate. So the concentration of glucose 6-phosphate inside the cell is almost certainly not the same as glucose outside the cell. And so we don't really want to measure glucose 6-phosphate inside the cell. Another note is the dermis has the largest volume of interstitial fluid, which has glucose, and plasma, which also has glucose. And so to do measurements for glucose is typically taken from the dermis. There's another topic to talk about, which is time lags. So that glucose came from the capillary to the vessels, into the interstitial fluid, and then into the cell. If you measure the glucose in the interstitial fluid, it's not necessarily exactly the same as that in the blood because it takes some time to diffuse through. There's been a lot of studies done on this, and there are things that change it. Chromium levels, there can be concentration gradients. It depends on how high level your glucose is, and temperature has a huge effect on all of this. So a lot of those studies depend a lot on skin temperature, which changes circulation. This is just a little note on that. So now that we understand the physiology, let's see how measurements are actually done based on the physiology. So on a finger prick system, a needle punctures the skin, it breaks a blood vessel, and out comes some blood to the surface of the skin, including some interstitial fluid. Then a secondary device comes along and wicks away that blood, and it reacts with an enzyme which converts to an electrical signal, and you get a glucose concentration from that. Alternatively, you see these minimally invasive probes that are out now that you can put into the skin and they sit there for a couple weeks. That probe pushes aside all that structure, the hair follicles and the glands, and it's usually a platinum wire coated with enzymes, and those enzymes react with whatever glucose is in there and create an electrical signal. So that's how the minimally invasive probes work. And then optically, if you want to measure it optically, which is ideal because it's totally non-invasive, you would focus the light into the dermis. You have to decide how how deep into the dermis to go, and how big the spot size is, and how vertically selective you'll be with your optical system to try to optimize your signal, because you're going to measure everything in there. You're going to see hair follicles and the vessels and glands. So it all gets collected. And so you try to optimize your optics to get the most interstitial fluid and plasma as possible. So a summary is finger sticks. They avoid organelles and skin structure by simply removing the blood out of there. You avoid them by taking the blood out. Now you don't have to worry about those things. And glucose specificity, in other words, the ability to see just glucose and not other things, is done by using an enzyme reaction. Minimally invasive probes, they avoid organelles by just pushing them aside. It's brute force. You just slam that thing in there and the stuff gets wedged aside because they're tough enough not to tear. It does cause some damage to the area when that's done. And so when the probe is first put in, sometimes the measurements aren't quite right. And that's why companies like Abbott and Medtronic are working on those types of issues. And for glucose specificity, it does it just like finger prick. It has an enzyme coated on the platinum wire that reacts with the glucose and creates an electrical signal. And those last about 2 weeks before they have to be pulled out and removed. Optical is tougher. Optical, it's going to see all that structure, but you can play with the optics quite a bit, changing the numerical aperture, and you can do something called confocal, which reduces its collection area to avoid light from the epidermis. You can also choose where on the skin you're going to measure. The skin is different all over the body. And so, for example, there's places of the skin that have very little hair follicles compared to other areas. So that would be a wise choice. As far as glucose specificity, well, you can't separate it with an enzyme chemically, but you do it mathematically by analyzing the spectrum that comes from the light and separating it out that way. Okay, I hope you enjoyed this tutorial and now understand how glucose is measured from the skin. Thanks.